Thank you. It's really an honor and privilege to give the Jeff Berger lecture this year. And as Josh said, Jeff was a resident at the Mass Eye and Ear between 93 and 96. And so Jeff arrived in Boston in July of 93, and I arrived in Boston in October of 93. I arrived at the Scapin's Eye Research Institute, which at that time was completely independent and separate from the Mass Eye and Ear. So there wasn't a lot of interaction. Now we are a part of Mass Eye and Ear, and that's corrected. But I did go back to his class, and I do know Catherine Colby very well. So I contacted her, and she, of course, had many, many fond memories of Jeff during the residency days. And the one thing that she said stuck out in her mind was he had an insatiable desire to learn more and more, and an insatiable curiosity. Plus, she also said she had a vivid memory of their graduation ceremony when they finished their residency. And so at the Mass Eye and Ear, there's an auditorium, the Meltzer Auditorium, which is the same now as it was back then. And Kathy's daughter was approximately the same age as your daughter. And she remembers when they were done, her daughter and Jeff's daughter playing and jumping off the platform at the Mass Eye and Ear. I also contacted several faculty members, and Joe Rizzo remembered Jeff, and we talked briefly. And then a couple days later, he came by and showed me a copy of a book that Jeff edited on macular degeneration that he still has on his bookshelf. So I would say, even though Jeff was there for a short period of time, he really had an impact on his colleagues and even the faculty at Mass Eye and Ear. So the title of my talk is Cellular Rejuvenation the Future of Ophthalmology? So what I hope to get across in this lecture, and I promise to stay on time so that there are questions, what I hope to get across is there is a huge potential and opportunity in cellular rejuvenation, but there are also problems or issues that have to be addressed to help translate this into the clinic. These are my disclosures. The only one that's important is the sponsored research agreement from Life Bioscience, which was completed 3 years ago. I'm not presenting any data from that part. This is important because Life Bioscience is actually this month or next month starting the first clinical trial on age reversal, and this is in the eye for glaucoma and NION. But I have not worked with the company for the last 3 years. So when I put this talk together, I really wanted to give 3 examples of cellular rejuvenation. The first has been published. The second 2 are unpublished data. The first is on retinal ganglion cells following aging, optic nerve crush, and glaucoma. The second is on retinal pigment epithelial cells following aging and oxidative stress. As hard as I tried to fit all 3 of these in, I could not get it within the time. So unfortunately, I'm not going to talk about reprogramming in myofibroblasts following induction of fibrosis. So in this project, fibrosis is a problem throughout the anterior and posterior segments of the eye. Reprogramming myofibroblasts, which help to secrete extracellular matrix and fibrosis. What we've shown in the cornea is that you can reprogram these cells back to fibroblasts. They are like original fibroblasts, but not completely. What we believe in preliminary results is that these cells retain phagocytic activity that can remove the deposition of extracellular matrix. And in this way, in the cornea, we've shown that you can actually resolve or dissolve a scar after it occurs. So I think this has a lot of potential for PVR and even end-stage gliosis in the retina to help restore that environment and make it susceptible for possibly cell transplants. Unfortunately, I can't show any of that data, but so to start definitions, what exactly is aging? I was 3 or 4 years ago, I was at a Gordon conference for the people that study aging. There were about 200 people there, and the researchers were all given a sheet of paper to write their definition of aging. What was clear then is there was no consensus among the people that study aging of actually what aging is, okay? So there was so many different. So my definition is a very basic biological definition, which a lot of aging research uses. It's a loss of cellular structure and function with time. When I say cellular rejuvenation, what I mean is restoring aged or injured cells to a youthful state. And so I like that definition. And you'll notice I didn't say age reversal, but it's implied with that. And the reason I don't like age reversal is because as soon as you say that, people think longevity, which is reversing age to increase lifespan. This is sort of a hot topic now. There's an editorial in Nature, and recently there was an article in the New York Times about this saying it's overhyped. I agree with those articles. I think it is overhyped. The articles also say there's a lot of opportunities still there, and I agree with that too. I do think it's very important to pursue this, to have cellular rejuvenation to treat age-related diseases. That's way more important than extending lifespan at this time. And the best place to study age-related diseases is in the eye. All the diseases, diabetic retinopathy, age-related macular degeneration, glaucoma, dry eye, all of these are age-related diseases. If you're interested in the field of aging, you want to get started, 2 reviews I listed here on the hallmarks of aging in cell are excellent places to start. They're 10 years apart. I would still read the earlier one as a reference for what's happening now. The second one's title is Hallmarks of Aging, an Expanding Universe. So this is a great place to start in aging research. I would like to say that in all the aging meetings I go, I'm the only ophthalmologist, and I would hope in the future, people in the audience would start looking at aging in the animal models of the eye. It's a great opportunity. If you go to those meetings, I guarantee you can get 2 or 3 new collaborations. I'm tired of being the only ophthalmologist at those meetings, so please start attending those meetings. So really this area started with Steve Horvath and others, but mainly Steve Horvath, that discovered the epigenetic clock. And so this is a validated biomarker of chronological aging. And so what you can see here is there's a predicted pattern of accumulated epigenetic marks with your chronological age. Okay, so you start out with very few epigenetic marks, and as you age, there's a pattern that corresponds very closely with chronological age. This is important because now we have a mechanism to show age reversal. You can't show you've rejuvenated cells unless you could quantitate aging. And so this biomarker was essential. Since then, this field has expanded. There are now epigenetic clocks, transcriptomic clocks, proteome clocks, and even image-based clocks. And just at Arvo, from Bascom-Palmer, there's an image-based fundus clock. Felipe Bascom-Palmer has shown that just by using AI and the fundus images, you can determine chronological age from an image. And this has also been done in non-ocular tissues too. So if you're interested in this field, Vadim from the Department of Genetics at Harvard has enormous numbers of papers on this field. It's really a very amazing and interesting field. I would look up his papers if you want to know more about that. So age reversal by epigenetic reprogramming actually occurs in nature. And that's we only understand that recently, but it solved a problem that has puzzled biologists for a long time. And so in development, you are given your lifetime supply of eggs, okay, during development before you're born. It was shown using the epigenetic clock that those eggs start to age, okay? And so they age, but it doesn't affect childbirth. So it doesn't matter whether you have a child when you're 19 or 40. There's no age-related effect contributed by your oocytes. And so with the epigenetic clock, we now know that the reason for that is the aging of the oocyte is reversed during gastrulation, okay? So once the egg is implanted at the blastocyst stage, there is an epigenetic reprogramming that resets it back to zero. And so this is why then it doesn't matter when you have your child, the embryo always starts from an age of zero. And so obviously you can imagine there's strong selective pressure for this type of mechanism, because if that wasn't the case, when you had your child, there would be a gradual accumulation of age-related problems that then would be propagated, and it would, in the species, actually fairly rapidly. This event has been shown in vitro. And so actually this was, if you remember, the first cloning of a large animal, Dolly the sheep. This was by single cell nuclear transfer. So the oocyte from a young sheep had the nucleus removed. They inserted a nucleus from an old mature donor. And what they were able to do is this induced reprogramming of that nucleus back to a stem cell. And then they cloned the sheep. It was clear then that the environment within that oocyte contained the environment to reprogram things. And so people look for it. Yamanaka, who won the Nobel Prize for this, discovered it was 4 genes that induced the complete reprogramming of mature cells back into induced pluripotent stem cells. So if you have prolonged expression of all 4 of these genes, you completely reprogram the cells back to induced pluripotent stem cells. So that's complete reprogramming. It's successful in vitro. There are reports that it forms teratomas in vivo because if you inject these cells, they respond to the surrounding environment and differentiate into teratomas. This has been solved now if you pulse briefly these 4 factors. So complete reprogramming requires extensive epigenetic remodeling of chromatin and DNA methylation to reset the somatic cell identity. And of course, the epigenetic clock is reset back to zero. So our lab with our collaborating other labs have shown that if we use 3 of those 4 factors and leave out the c-MYC, we can induce epigenetic reprogramming, which is more restricted just to age reversal, okay? So this c-MYC we left out because it's an oncogene and also is associated with advancing age. So if you just use 3 of the 4 factors for a short duration, you don't change the cell identity. But what you do change is you reset the epigenetic clock backwards. Doesn't go all the way back to zero, but you do reverse it backwards. There's no loss of cell identity. As I said, no tumor formation, at least within the experimental systems we've studied so far, and it's successful in vivo. What I'm going to tell you today in the unpublished data is this is cell type specific, and there are different effects on neuronal versus non-neuronal cells. So to test this, we first expressed these 3 of 4 Yamanaka factors in retinal ganglion cells. So we did an intravitreal injection of AAV targeting the retinal ganglion cells. So I'm only going to summarize this because this was published in Nature now 6 years ago. It was a collaboration between David Sinclair's lab my wife Meredith and my lab and Shigan He's lab at Harvard. And it was the thesis project for Yan Chen at Harvard. And so while this was an enormous project, he really spearheaded all of the experiments in this study. And we looked at 3 different systems, Shigan He's lab, optic nerve crush, and in Meredith's lab and my lab, we looked at aging and glaucoma. And so important to recognize is we looked at optic nerve crush and glaucoma in young mice and only reversed aging effects in the old mice. That illustrates the close association between injury and aging. And so injury actually now has been shown to accelerate aging. The reason for that is if you injure a cell, it induces epigenetic changes that would occur normally later on in aging. And so that's the association between injury and aging. There's actually been historically theories of aging that predict aging is the accumulation of repeated damages of tissue over your lifespan. So this is published, so I'm just going to briefly summarize this data. For optic nerve crush, if you activate the OSK factors after you crush the optic nerve, in adults, there's no regeneration of the axons. If you activate the rejuvenation factors in retinal ganglion cells, now you can see significant axon regeneration. We know quite a lot about axon regeneration. So this just summarized the results. All 3 of the OSK genes were required. This is important because it showed that you had to deliver all 3 genes at the same ratio into cells. If you use vectors, individual vectors, you never achieve that. This was important because it was hard to package all 3 genes into one vector. It decreased the methylation age by the epigenetic clock, didn't induce cell proliferation. This is important because it demonstrated the epigenetic basis for this. If we knocked out TET1 and TET2 with our demethyltransferases, we eliminated this rejuvenation effect. It was also dependent on TDG. It increased axon regeneration not only in young mice, but also in old mice. And the axon regeneration was cell autonomous. We haven't actually proved this, but we tried to show that it was cell intrinsic and not dependent on the surrounding environment by expressing it in amacrine cells. And when we did that, we didn't induce regeneration. In aging mice, we showed that 4-week treatment of retinal ganglion cells restored both pattern ERG and visual acuity by OMR in old mice. Importantly, the transcriptome and the methylome was restored to a youthful pattern. And the epigenetic age, again, was reduced, also required these epigenetic TET1 and TET2 genes. And also, it activated components of the polycomb repressive complex, okay? So for glaucoma, we know the least about it, but we did show following a 4-week treatment of retinal ganglion cells, a restored pattern ERG and OMR. Importantly, this was after you induce the damage by elevated pressure. And so this is important for translation because it showed that you could induce injury in retinal ganglion cells, show this by reduced visual acuity, rejuvenate dysfunctional but not dead cells, and restore the visual function. Okay, so at this time, our idea of what the mechanism behind this is in young DNA, there's low levels of methylation. As you age and experience injury, this level of methylation increases, and this is associated with impaired function and regenerative capacity. When you reprogram cells and rejuvenate them, what you're doing is you're activating PRC2 and EZH2. This activates the demethyltransferases. And so these go along and specifically remove the epigenetic marks that are associated with age. They don't remove all types of epigenetic remarks. They're fairly specific for the age-related ones. The mechanism behind this is unknown. This then restores it back to the low methylation level, restoring the function and regenerative capacity. Okay, so that was published since our report in 2020. This has now been shown in many systems outside the eye, in the optic nerve for multiple sclerosis, in the brain, skin, heart, knee. So it's been reproduced, showing there's a reduction in the epigenetic clock and also restoration of function and restoration of the transcriptome. So in addition, there are many labs working on other methods of rejuvenation. So these are associated with people after Yamanaka showed you could induce iPS cells. People looked at a variety of other methods on how to do that. And they used chemicals, they used small molecules, and they found that you could induce reprogramming. And so all of these different types of methods are now being used in a transient epigenetic approach to rejuvenate cells. This hasn't been published, but I know there's several laboratories working on really exciting idea that you could just use metabolites to trigger reprogramming. Okay, so actually when we were doing this and before we published this, and I was doing the experiments, I was trying to convince myself that this was actually real, okay? So reversing age seems kind of, I mean, I wasn't in the aging field, and when I look at this, aging has so many different, manifests itself in so many different ways. To me, it didn't make sense that there was one central mechanism that you could target and you could reverse aging even when people were aging by different mechanisms, okay? So it was my data, it was positive, but I wasn't convinced. And so I was thinking, well, what would convince me? And so I thought if I could see a cell that looked old and became young, that would convince me. Can't do that with retinal ganglion cells because I could show you a picture of a retinal ganglion cells from a young mouse or young person. Old retinal ganglion cells, they look the same. Retinal pigment epithelial cells, by contrast, have age-related changes not only in function, but also in morphology. You can look at a mouse RPE cell from a 2-year-old mouse, and it's very different from a young mouse. So I thought, if I do that, it would convince me. So I went to Magali at the Scapes Institute studying RPE, and I said, I got this great project. Work together. We're gonna, show that you can reverse cells. It's going to be easy because we already got this other paper. It's going to be no problem. Slam dunk. It's going to be terrific. So the idea was I really wanted to just see them change. But the other rationale was that this was not a neuronal cell. So this is neural ectodermal derived, but it's not a neuron. And the most important thing is age-related oxidative stress contributes to AMD. And we found evidence in the retinal ganglion cells that reprogramming and rejuvenation, one of the main targets was oxidative stress. And so we set out easy experiment because instead of targeting retinal ganglion cells, we do a subretinal injection, same vector, express it in RPE cells. First result, fabulous. So this is visual acuity by OMR, young mice, old mice. These are 17 to 20-month-old mice. You express OSK and you increase visual acuity. And when we looked, there was a decrease in mitochondrial size, a decrease in RPE lipid deposits. you could visually see a change, okay? So in addition, when you inject it, there's an injection site, and we could see a change in the injection site, but not the surrounding tissue. So there was an internal control showing this change in morphology. So remember, I wanted to see it, and so I saw it. Okay. Then, unfortunately, Magali decided to go to Novartis, and her lab was shut down. So I wanted to finish this project. So Yan Chen, who now graduated, started his postdoc in Jonathan Wiseman's lab at the Whitehead Institute, and I was also working with Jason Monrostro. I went to Yan Chen and said, okay, all we have to do is finish this. This is going to be an easy paper, high impact. It's going to be great. So that was 6 years ago, and we're now just about to publish the paper. Now, so the reason I love this project, because not only will it illustrate the problems with rejuvenation, but it's also what I think is the most fun research project. And that is, we think we have it figured out, and then we do experiments, and every experiment we do doesn't tell us what we expect, okay? And so if your postdocs typically, or graduate students, you come in, don't tell your mentor the experiment didn't work. So the experiment doesn't work when you like drop the plate on the floor. It's a technical problem. But if you set up your experiment, the data you get is the data you get. It's not that it didn't work. It's you're not getting the results you predict. This project, for the years, we were not getting the results. I say it was fun because it's in retrospect, okay? So there's a happy ending to the story, but at the time, it's frustrating. This actually turned out to be good because it's sort of that project that you're trying to figure out, what's going on. Yan Chen caught the bug. I was fortunate to get Jonathan Wiseman interested in it, and this turned out to be Yan Chen's postdoc work. So the first problem occurred was Yan Chen repeated what Magali and I had done, showed the positive effects in morphology, the positive effect in visual acuity. Next experiment is to reverse the age. And here's the transcriptomic clock. Old mice, high age, young mice, young age. And you can see we reprogrammed them. And instead of reversing the age, we accelerated the age. And we were looking at it and said, well, it's not that much of an increase, but it was significant. And, we were saying, okay, we did a bunch of other clocks and got the same result. But then we looked at the transcriptome results. And so this is the transcriptome of the RPE cells, and it's separated into pro-aging transcriptome changes versus rejuvenation. I was pleased to see, as an immunologist, it affected inflammation and metabolism, but you can see this is not what we expected or wanted. We then went on to look at A, B, and C ERG responses. Those actually went down. We looked at microglia activation, and actually we got massive movement of microglia to the outer retina in the RPE, which is never a good sign. It's an indication of some sort of dysfunction in RPE cells. The final nail in the idea was we looked at bulk RNA-seq responses. And so these are purified RPE cells. And so we looked at RPE maturation functional markers. And so this is the negative control. So in iPS cells that are differentiated, obviously we're not going to express markers for RPE cells. They do express proliferation markers and subcell markers. If you look at young RPE cell, they have RPE maturation and functional markers shown here. We looked at this because this is a control for malignant transformation, which we don't want to get. If you look at old RPE cells, you can see now, as you'd expect, they're losing some of the markers and function. this is the transfection control, but when you add the reprogrammed ones, this was definitely not cellular rejuvenation. This is driving cells in the wrong direction, okay? And so this is not what we wanted to see. You could argue there's some positive results because this was not associated with proliferation or stemness, which would have been tumor formation, but definitely you just can't make the argument that you rejuvenated these cells. So we then looked at a bunch of RNA-seq and attack-seq data from RPE cells with and without OSK. And we found, maybe not to our surprise, but there was extensive chromatin remodeling in the RPE cells. So it opened up 40% of the genome. Okay, so the chromatin in differentiated cells closes off except for limited areas for transcription. This exposed 40% of the genome to open up a huge number of different gene network pathways. That was associated with changes in transcriptome of 6,000 genes. So we thought, well, is it possible to separate out these positive effects? Because remember, I originally said if I could just see the cells change, I would be convinced. And I saw that, but actually what I was seeing didn't match up with the data. I still thought there were some positive effects. Could we separate the positive from the negative effects? So we wanted to think, let's look at the downstream targets for OSK, and maybe those gene targets we can separate out positive from negative. But there's 6,000 gene changes. So where do you start? So fortunately, this is exactly what Jonathan Wiseman's lab is interested in, and that is doing large-scale screens to separate out these different responses. To do that, you need to have a assay screen. And so we picked a screen for oxidative stress. We thought this was important. And Yan Chen for CSIS did two types of screen. I'm going to just briefly show the functional genomic screen. I don't have time for this whole genome screen. But so this is the idea behind this is we're going to take OSK treatment and identify downstream gene targets that are activated. This is even cooler than that. It said, forget about OSK. We're just going to take RPE cells and screen all the open reading frames in the genome, and we're going to move it up and down, so CRISPR amplification, inhibition, and see how it affects oxidative stress. This revealed a gene that was never expressed in RPE cells, but is expressed in the heart. And interestingly enough, the function in the heart was developmental, but it was still expressed as the adult. But for whatever reason, the person that cloned this gene, they could never figure out what the effect was in the adult. And so this screen showed that it had enormous protection from oxidative stress. And so I wish I had time to go over this, but so I will just going to focus on the downstream assays. So this is a complicated slide, but it's just to do a screen, you need an assay. We used ARP19 cells, and so RPE people that study this knows that these aren't great. They're not like human RPE cells. We had to use these because when we used iPS-derived RPE cells, we couldn't transfect them. They're resistant to transfection. So this is just an assay showing we differentiate RPE cells. We then give them this huge oxidative stress, and normally that kills them, okay? And so we're then going to screen genes here for resistance to this oxidative stress. And for OSK, we turned it on. And what I will show you is when you turn it on, you get protection, really good protection, And that's shown here. So you stress RPE cells, you kill them, you express OSK, you protect them. This is the on-off. So if you remember what I said before was that you had epigenetic dependence on TET1 and TET2, that is a memory response. So you put in the epigenetic signals, you remove OSK, those cells continue to behave because of that imprinted epigenetic signal. We weren't seeing that here. So we did a bunch of experiments where we removed TET1 and TET2, and it never budged this. Okay, so right away now, this is no longer epigenetic reprogramming, it's something else because it's not dependent on epigenetics. So very complicated. This is the Jonathan Weisen lab functional genomic screening part. Essentially, here on the left is what they did is they took the RNA-seq data from human RPE cells plus or minus OSK and mouse RPE cells plus or minus OSK. Like I said, there's 6,000 differences in human, 1,000 in the mouse. They shared 250. 250 still is too large to screen. So this was then combined with a sensitivity to oxidative stress. 7 candidate genes then were cloned into this screening assay. And what we found was, as you'd predict, OSK is like super good at protecting these cells, but we found one gene, GSTA4, which was pretty good. Okay, so we now out of the 6,000 genes, we have one we're going to study. And so the first thing we did was, did it reverse or accelerate aging or not? And now when we expressed GSTA4, we did get age reversal. And the important thing before was rejuvenation is restoring youthful markers. And so this was the experiment I showed you before in RPE cells. This is young RPE cell youthful markers, goes down a little bit in age. And now, instead of reducing those markers as OSK did, GSTA4 did a great job of restoring markers found in young RPE cells. And so, we did other experiments. I'm just going to summarize. So what is actually going on here? So we did a bunch of experiments to prove that this is downstream. And so we did something called cut and run to show that the OCT4 of the OSK binds the promoter of GSTA4. So this activates GSTA4. It's not epigenetic dependent because when GSTA4 is removed from the promoter, the gene is shut off. So this is very different from the epigenetic reprogramming I talked about in the beginning. It produces GSTA4. So how is this protecting? So during increased oxidative stress, increased ROS levels, there's lipid peroxidation products, which an important one is 4-HNE, and it links oxidative stress to cell death. And so remember, our screen was measuring oxidative stress and cell death. So we found one of the targets of that, highly toxic. So this, when you have oxidative stress, this is actually one of the things that kills the cell, okay? It can be mitigated though. And if you have glutathione, it's a detoxifying agent. Glutathione binds 4-H&E and forms this complex, which is not toxic and can be cleared. Do you remember Magali? For the 6 years she was in Novartis, we told her all about this data, even though she was in industry because we trusted her. But after we told her this, she started taking glutathione supplements. I don't recommend that, but Magali told us that what GSTA4 does is actually it's a transferase that catalyzes this reaction. Okay. So Magali realized, okay, we're expressing this. What if you don't have enough of this? So if you take the supplement, she doesn't have the gene therapy yet, but she's just hoping maybe this will help. But if you have all of these, one part was when we were doing our experiments, we predicted that most assays try to reduce ROS, okay? And so we were puzzled in our experiments because we were getting protection, but we weren't reducing ROS. So the reason is the effect was downstream of that. And so this is a protective mechanism that allows cells to rejuvenate, function normally, even in high levels of free radicals. Okay, so promise to stay on time. And so I hope what I've conveyed is some of the really exciting potential there is for cellular rejuvenation, but it is not a simple process. Therapies that reprogram or remodel 40% of your genome aren't targeted, personalized medicine. I mean, it's just opening up a huge number of pathways. Here we've shown a program or a method to separate out positive from negative effects. And we think this targeted approach that will specifically target parts of the rejuvenation process has a lot of translational benefits. So with that, I'd like to acknowledge all my collaborators. This is present and past members of the laboratory and my funding. And thank you.